what I want to run through here. So on the study guide, when you look at the study guide, that is linked at the bottom of the blood unit page because for some reason it wouldn't let me link it from the schedule. But you'll see it there. I put it in all caps, Unit 1 study guide. So for the immune function, all you're really doing is the leukocytes. I should take this off actually. Leukocytes, you do want to know the leukocyte functions and categories. So I'm going to run through leukocytes. You don't need to put a whole lot into this other than knowing the categories of leukocytes and what functions they play, what roles they play. After that, we'll go through hemostasis and look at the 4 or 5 steps in blood clot formation. So leukocytes, or white blood cells, are larger than erythrocytes with a very prominent nucleus and often identified under a microscope by the shape of that nucleus. So they use the bloodstream as transportation. They are a kind of red blood cell made in a very similar fashion from erythroblastosis, but this is leukoblastosis. And we have stem cells in the bone marrow that make both red blood cells and white blood cells. So as immune cells, they don't generally perform their functions within blood. That's just a transportation device. But they do adhere to walls of blood vessels, and they squeeze between the endothelial cells to enter the surrounding tissue. They're kind of oozing in and out of the transportation system, exiting here and there to check your tissues and then get back in. Leukocytes are divided into 2 basic categories: the granulocytes, which contain cytoplasmic granules. They have a grainy appearance. We'll look at that. And they are released when activated. Agranulocytes lack those visible granules. So here are the granulocytes. This is a main category that we want to be able to identify. So the way I think about this is look at the suffix of these: neutrophil, eosinophil, basophil. So these phils are all filled with granules. And you can see that grainy granular appearance. So that's one way to remember that. Neutrophil, eosinophils, and basophils are granulocytes, that is, cells, sites filled with granules, because that's what they look like under the microscope. So these are the classes of leukocytes, and you can get a description there. I will not have pictures of these on the test. It'll simply be asking you for basic identification, again, by which ones are granulocytes and what is their function. The others are agranulocytes, and these do not have the word fill in them as the others do. Although this is a fuzzy appearance, makes it look like it has granules, but that's just up close. It's large. So monocytes and lymphocytes play large roles in immunity, larger roles in specific immunity. And we'll discuss that with the immune system next week, not on this test. But you do want to know that lymphocytes and monocytes exist, of course, as a kind of leukocyte that is agranulated, as opposed to the phils, neutrophils, eosinophils, and basophils. All right, you can go through these slides then, and again, simply just understanding the basics of what a granulocyte does, knowing what a neutrophil is most common, and it is an active phagocyte. So neutrophils are the most common, walking around, going in and out of your blood vessels, phagocytizing. If you don't remember what phagocytizing means, it's cell eating, right? It ingests and destroys bacterial cells. You have neutrophils. Whoops. Chemotaxis is what I was just describing, the moving around in and out. So we'll talk a little bit about injured cells releasing chemicals that attract neutrophils when we get to the hemostasis part. But again, more of that will be in the immune system, general immunity. I'd just be reading these slides to you, so I'm going through seeing if there's anything special I want to explain, but I think these are all pretty self-explanatory for now. You want to liken the eosinophils to parasites. I should say that much. Anyway, basophils to allergic reactions, mediate inflammation, swelling that is, in attempts to protect the body. So those start on slide 15. agranulocytes, the monocytes, and the leukocytes. Then lymphocytes and monocytes are now spoken of. Again, I'm going to go on. We'll talk about B cells and T cells and lymphocytes with the immune system. So a lot of these you don't have to pay a lot of detailed attention to, although of course for your professional lives you want to. I'm just saying I'm not going to hold you highly responsible on this exam for it, other than the categories of granulocytes and agranulocytes, and then the basic function of each of the 5. Again, neutrophil, basophil, eosinophil, and then the monocytes and lymphocytes. If you know the basic function, you're good. This can all be accomplished with probably 2 multiple choice questions. That is, all of the following do, or which of the following do, and I would give you like 4 functions of white blood cells, and I would pick a lymphocyte and ask you which one is a lymphocyte. Or I might say, a neutrophil and give you all the functions, and you have to identify which one's a neutrophil. So again, not heavily tested, but of course you need to know at least a good number of these, maybe, so you can pick the right answer by process of elimination. All right, I want to move on. It's been 8 minutes already to slide 30. This is where I'll spend about 10 minutes, maybe less, hopefully. So when a blood vessel is severed, a number of processes aid in wound closure. This is one of the most marvelous feats and processes in the human body. There's local constriction of blood vessels due to local neurogenic causes, which we'll see, that cause a vascular spasm. When I say neurogenic, what I mean is the nerves that innervate the blood vessels, that stimulate them to contract. So you might recall from anatomy that you have smooth muscle, skeletal muscle, and heart or cardiac muscle. So smooth muscle is the muscle lining vessels, and that we have no control over. That is not a voluntary muscle. And so this is what I mean by neurogenic. So nerves, if they are stimulated by way of injury, they may go off and cause the smooth muscle to constrict, causing a vascular spasm. Right. So then there's if it's a little bigger, and these that kind of damage occurs all the time in blood vessels. You don't have to be playing a basketball or football game to have internal injuries. I mean, these are just sometimes the harsh molecules from the foods we eat and passing through can cause these micro damages. And vascular spasm is one way to slow down blood flow in that area for repair. All right, well, the next stage would be the formation of a pure platelet plug. So in the case of small wounds to capillaries or very small blood vessels, the pure platelet plug is often sufficient to close, again, small wounds, even without a blood clot. Does not need to coagulate. And we'll look at pictures of each of these. But of course, the next step is coagulation, and that's the formation of the clot itself. And that involves platelets, fibrin, as we saw in lab, holding up red blood cells themselves. So that's a whole team approach right here. And then, of course, once that clot is formed, on the back end of that clot, the endothelial lining of the vessels begin to heal themselves. And once they're healed, we don't need this clot anymore. It breaks off, and we have clot retraction and thrombolysis. We got to break up that clot. It cannot stay a clot in your bloodstream. So let's go through some slides and look at this. But this is what you want to know for the test. This is the basic sequence of what you want to know. Study the diagram. So here's the rip in the vessel wall. Chemicals at this point are released. Also, the cell contents are exposed, and so there's a lot of collagen. And if red blood cells or platelets come into contact with the collagen, boom, that sets off a domino effect of chemical reactions. Blood leaks into the extracellular fluid, vessel spasms, and leaked blood increases tissue pressure, both of which decrease blood flow and blood loss. So by constricting, we slow down the passage of blood, and by slowing down passage of blood, we slow down blood loss. So that is how vascular spasm works. That's kind of like first level of homeostasis by hemostasis. Don't mix those two words up. Platelet plug formation. Vessel injury exposes collagen fibers, as I said, in the endothelial wall. So if you recall, again, I don't know everything in your particular anatomy. I've taught anatomy and physiology. I've not taught just anatomy here, so I don't know. But this is at microanatomy here in the blood vessel walls. This is your endothelial lining. So these are epithelial cells, particularly squamous, simple squamous layers. So here are some interleukins or cytokines, just chemicals that are released by injured cells that kind of recruit help. They activate platelets to come to the point or area. We have these von Willebrand factor, which also then binds platelets and causes and activates them to stick to the exposed collagen. So platelets are pretty smooth and run through the blood on a regular basis, but once activated, I mean, it's amazing. They morph into these really sticky little things, and they just attract more and more of each other. So these are activated platelets, and they release the contents of their granules to say, hey, I've been called, and now I'm calling you. Throw me esquinas, we call it. Give me corners. Call some help. So everybody comes, and when they come, they send a message to call more. And those come, and they send messages to call more. So all the platelets release their signals. So the contents of platelet granules activate and attract more platelets, which aggregate to form the platelet plug. All right. Third step is coagulation. This is a pretty complex step, and we won't go through everything, and I won't have you memorize everything, but we will go through it just so you can appreciate it. So then factors 10A and 5A. So let me just say about these factors, there's 1 through 10 factors, but they're not named according to the sequence or order in which they work. They were named in the order in which they were found or discovered. So unfortunately, there's not a real rhyme or reason at this point for learning them, as you'll see here shortly. Anyway, so factor 10A and 5A start off along with the calcium as a signaler for prothrombin. Prothrombin. That word pro means before. So thrombin would be the protein. Pro is before the protein. So this is a prothrombin activator. So an activator has to be activated, and that converts prothrombin into thrombin. It's a little confusing. So here's prothrombin, here's thrombin. It's the same protein, but in an active form. But what activates the prothrombin activator? 5A and 10A. So this is the prothrombin activator, and boom. That's going to be important because there's a couple different ways this might occur. We'll talk about briefly, but both what we call intrinsic and extrinsic pathways will end right here, or I should say join right here at prothrombin, thrombin, and from this point on it's one single pathway. I'll hope to unravel that confusion here in a little while. So anyway, now that we've converted prothrombin to thrombin, thrombin turns fibrinogen into fibrin, which now glues the platelet plug together. And there's this. So again, fibrinogen is always present in the blood. It's one of the primary 3 proteins circulating in your blood. If you saw earlier in the chapter, you have albumin. That's the highest quantity of protein made by the liver. That albumin kind of serves as an osmotic. It serves in blood pressure. It serves in a lot of purposes, honestly, but one of the main purposes also we'll talk about later is to keep that osmolarity up in the blood. So there's albumin. We also had gamma globulins and immune proteins circulating in the blood plasma. And the third kind of protein is this fibrinogen. So kind of like prothrombin is before thrombin, fibrinogen is the progenitor, generates fibrin. So fibrin is the active form of fibrinogen. This is the one we saw working with our own eyes in the lab under the microscope. It associates then with the protein. So here's a nice scanning electron microscope image of red blood cells trapped, erythrocytes trapped, platelets trapped in these fibrin threads. Here's a leukocyte moving around, eating up the junk that might be coming in or out. But that's a pretty amazing shot right there, and that's what it looks like. A bunch of silly string attaching all those things together. All right, then there's a clot retraction. The coagulated clot once this has been glued together and the body begins to heal itself, as you see here, it's closing, so the endothelial lining is repaired. Now serum is squeezed out of the clot by contraction, brought closer together. Now this will have to be dissolved by other enzymes. And here's platelets as well, free now, and this is thrombolysis. So endothelial cells release tissue plasminogen activator. The plasminogen activator activates plasminogen. Duh. Think about that one. Plasminogen is inactive. Whenever it has that ogen or pro before it, that means it's an inactive form of the protein. The active form will always end with an IN. Plasmin, fibrin, thrombin. So plasmin degrades fibrin and the clot dissolves. Whew. So here's a picture of the whole thing. You can go through again slowly and look at it. Just want to remind you, this is a positive feedback mechanism. The response enhances or exaggerates the original stimulus, says more of it. It's to seal off the whole. It has an amplifying effect. We see this. Here's just two other examples from chapter 1. Just another diagram, a summary diagram. The stimulus is the chemicals released during the blood cell wall break. Platelets adhere to the site and release chemicals that attract others. And that's the positive feedback right there. So I've been initiated, and I'm going to work, and I'm going to call more and initiate more, and more and more and more brings more until this is sealed and the original stimulus is gone. Almost right away that plasminogen is secreted, but this is just such a force to reckon with that really until this signal that says make more is gone, then it's not going to. So this part is a positive feedback. And plug forms. There's that picture up close and personal. Here's a picture of hemostasis, summarized vascular spasm, platelet plug formation, and then coagulation. That's with the fibrin. Fibrin is key here. Here's some collagen. So, clinical disorders of clotting. There's a few. You've heard of hemophilia. Hemophilia, most common type of hemophilia, classic hemophilia, we're missing factor 8 right here, V3. So here's where it begins. This is where it ends with fibrin. So there's two ways to get to this prothrombin activator. One's called the intrinsic pathway, which happens when red blood cells undergo trauma, damage, or they come into contact with the surface of something other than their cells, like collagen sticking out from an endothelial cell. If an endothelial cell lining has been ruptured or torn by some harsh molecule or foreign object, when a red blood cell touches that or hits it, it's activated to clot. Well, in order to get fibrin, you need thrombin. In order to get thrombin, you need to be activated by the prothrombin activator. To get that, factor 10 has to tell you, and work your way up the chain. So in hemophilia, you're missing factor 8. So none of this down here can occur. Therefore, you're not clotting, you're a bleeder. And another lesser form of hemophilia, it's factor 9, which is right above it, is the one that's missing. So and these are gene mutations. These are genetic defects. This happens to be a sex-linked trait as well.
A lightning warning has been issued.
Lightning warning. All right. So I think that's a pretty good little introduction. Again, so blood clotting is complicated. I won't have you memorize this, of course. The book goes at length about the intrinsic and extrinsic pathways. The extrinsic, real quick, I didn't say, is tissue trauma and releasing thromboplastin and things into the vessel, which acts as a signal for clotting. So there's intrinsic, which is trauma to the red blood cells themselves, or extrinsic, which is damage to tissue. So in each case, you got two different sources. They'll come down by two different pathways. but they'll converge at the prothrombin factor, and these are all the same after that. Anyway, you can look to your book for more details, but I won't ask you for a lot of those details. If you have these 4 major steps, understand these primary steps right here in hemostasis, you should be good to go.